So good morning to all of you. Today our topic is phylum Porifera, the first phylum of kingdom Animalia. Porifera are also known as sponges, and they are the simplest type of animals in the kingdom Animalia. The study of Porifera or sponges is known as parazoology. So parazoology is the study of sponges, and the scientist named Robert Grant, he coined the term porifera. So two important points: parazoology is the study of sponges, and Robert Grant coined the term porifera. Okay. So this is the simplest type of structure of porifera, and as I am telling about the porifera as the simplest type of structure because they do have cellular level of organization, and phylum porifera is the only phylum in kingdom Animalia has a cellular level of organization. No other phylum in the lower animals has cellular level of organization. That's why they are the simplest type of animals in this kingdom Animalia. So let's discuss about the structure of porifera or sponges. Here you can see the simple structure of porifera or sponges. Uh, you can see they are actually all of the sponges of porifera, they are aquatic in nature and mostly marine, means they are found in sea water, ocean water. So these are the porocytes, and they are also known as ostia. Ostia and osculum are kind of two inlets and outlets. That means water flow inside the body and water flows outside the body. So there are two inlets. So there are actually two pores, or you can say a kind of openings which facilitate water to flow in and to flow out. So this porocyte, which is also known as ostia, they serve as they serve as the conductor of water. Actually, water flows in through this porocyte or ostia. The similar structure of porocyte is, you can see over here, these are incurrent pores. The function of incurrent pores and porocyte is same, and both of these parts are termed as ostia, and water flows in through these incurrent pores and porocyte. One more structure in the pink color you can see over here, known as spicules. The spicules actually, they are calcified and they provide support to the body of sponges, or you can say that they form the skeleton system of sponges. And beside the spicules, another non-cellular matrix which is known as mesohyl, which is present within these gaps or spaces. Mesohyl is a non-cellular matrix which also provides spores, or cells of these sponges are suspended in the non-cellular matrix known as mesohyl. MESOHYL. So spicules to form skeleton system, spores body, porocyte also known as ostia. Similarly, incurrent pores, ostia, water flows inside the sponge body. Okay, and when water gets inside the sponge body, along with food particles, gaseous molecules which are very necessary for sponge to survive, they enter through these porocytes, and when the porocytes end, this structure is here known as choanocytes. These choanocytes actually are responsible for digestive process, mainly digestive process. So these conocytes are present over here to capture, to engulf food particles and also to capture gaseous molecules which are necessary for the survival of sponge body. So these conocytes are present over here to receive food particles, gaseous molecules from this porocyte where water is flowing inside. And after capturing the food particles and all the necessary elements from the water, now there is no function of water is left, so water has to flow outside the body, which is through this osculum. There is an opening which is known as osculum, and from this opening which is known as osculum, water flows outside the sponge body. This is the outer layer which is epidermis. These are the cells which are known as pinacoderm cells, which are similar to that of epidermis, okay, forms an outline of the sponge body. And this inner cavity is known as the spongiocele, which is internal cavity of sponges. The most important part, which is coenocytes, I have drawn over here. This is coenocyte, this structure I have drawn over here. This coenocytes is consisted of the various structures. You can see over here amoebocyte. This is amoebocyte cell which has function in making the structure of sponges, in reproduction, in regeneration when it gets any injury, so regenerate any part of it, and also in digestion because it also helps in engulfing the food particles. And here in this part, phagocytosis takes place, means engulfing of food particles take place in this part. And these are collar cells. These collar cells actually they force water through the sponge body. जैसे ही पानी पोरोसाइट के थ्रू अंदर आएगा, ये फोर्स करेगा पानी को इस तरह से बाहर निकलने के लिए, ठीक है, यानी एक तरह से ऑस्मो रेगुलेशन को ये मेंटेन रखता है, द कंसंट्रेशन ऑफ वाटर और मॉलिक्यूल inside or outside the body. And this flagellum also helps in actually creating the current inside the sponge body. So it's all about the structure of the phylum, structure of sponge. Main points are coenocytes, digestive property, porocyte or ostia, water flows in, spicules form skeletal system, spongiocele is internal cavity, osculum water flows out. And pinacoderm is similar to that of epidermis, is outer layer of cells. So as these characters, all are aquatic, mostly marine. Cellular level of organization, that means the simplest type of animals. Sponge skin is composed of two special cells. One is exopinacocytes, outer ones. Exopinacocyte cells and basipinacocyte cells. These are two specialized cells which form the skin or outline of the sponges. These are the distinct features which differentiate sponges from other animals or the other animals of the other phylum. Okay, तो ये बाकी phylum से जितने भी जो animal kingdom में animals हैं अलग अलग phylums में उनसे इस porifera को differentiate करता है on the basis of these distinct features. These are coenocytes. They are only found in porifera. Coenocytes, water canal system, ostia, osculum, ostia, osculum. Organic and inorganic skeletons, spicules and mesohyl, as I have told you, mesohyl, non-cellular matrix. Totipotency means ability of any cell to form all new cell or any part of cell. And plasticity, the flexibility or inside the water or in the tides of ocean. These archaeocyte cells, they act as stem cells, and as you know, stem cells has capability to change into any type of cell of the body. So these archaeocyte cells would behave like this. Parazoology, study of sponges. Robert Grant coined the term porifera, and the reproduction or asexual reproduction takes place through budding and fission. Okay, simply the division of the parent cell, and sexual reproduction by producing sperm and ovum, and the sperm and ovum actually derived from the structure archaeocytes or choanocytes. In archaeocytes or choanocytes, actually gametogenesis takes place in archaeocytes and choanocytes. That's why sexual reproduction is derived from archaeocytes and choanocytes. Then, on the basis of the canal system, uh, actually we classify sponges whether they are simple or complex. So there are, on the basis of a canal system, sponges are either ascon type, cycon type or leucon type. Ascon type of sponges are the most simplest type of sponges exists. Cycon, they are complex sponges, and leucon type of the sponges are most complex type of sponges, or you can say they are most primitive type of sponges, and leucon type are they are most evolved type of sponges. For example, in ascon type, leucosolenia; in cycon type, Scypha and Grantia; in leucon type is Spongilla. You can see the biggest type of the sponge. This is the structure of actually Spongilla structure is there. So it's all about the phylum Porifera. I hope if something is left, you can take the help of internet and search more about phylum Porifera, but do revise whatever I have drawn over here and the points I have written over here. And in the next lecture we will discuss about ceiling traits. Thank you.